me start recording this real quick. No, I was just reading an email saying that the old attendance link wasn't working, so I'll have to give you guys a new attendance link. Make sure we got that under control. But okay. So what are we doing in lecture this week, or what do we need to work on? Is there some important topics that we think are worth going over? You guys can feel free to give me a chat real quick just to make sure. It looks like we have 19 people. That's great. That's a good turnout. We should get a good conversation tonight, hopefully.
I want to say something.
Yeah, go ahead.
Okay, so I see a lot of people complaining about how they're not passing the exam that they took already, exam 1. And it's just overwhelming, starting to get so overwhelming. Like, no matter how hard they study or we study in general, it's not working out.
So the lecture.
Yes.
So we need to switch up our study approach, it sounds like. Anything, Meredith, I just saw your metabolism chapter. So what are we doing? Like, what are you hearing that people are doing that they're not succeeding in? Their study approach? Are they just staring at the notes, or are they studying
They have tried almost everything, including flashcards, which I tried it myself. It works very well in every other class, but when it comes to this, like biology, it's not working.
So I've told my students in lab that I recommend writing everything out, and let me show you what I'm talking about. This is something I've been working on at my work, and I'm going to flip it up for you. This is how I do things. All right, and this is sorry, my microphone muted, but this is how I've gotten through school and this is how thick it is. Okay, and it's taken me, this has been months and months of work, but it's hard work and it takes a lot of time to do. So what I recommend doing is recording all of your lectures and replaying your lectures back and writing it down, all the important parts. If you are confused, then you have to go back and go through that material again. And the more passes you get on the material, greater likelihood that you will understand it even a little bit deeper. Okay, so these spotlight sessions are supposed to help that. There's supposed to be another source to talk about topics that are hard to go over. I know Meredith mentioned metabolism. Jennifer made a comment, which is great. However, topics are usually the best part. And then once I get certain topics like metabolism or channel membranes or transporters, then I can go into detail. But from a systemic point of view, I would definitely recommend recording the lectures so you can go back and listen to them and then write it out what these organelles do or what the metabolism is, and you can write it out in your own words, and that'll help you retain it. Flashcards is a memorization technique that, yes, it works in the lower level classes. However, when you get into the 300 and 400 level classes where they start manipulating things, you might have learned it that the Golgi apparatus does this. Okay, well, if you take the Golgi apparatus out, what is the most likelihood that something's not going to occur, or what it will affect? Well, if you take the Golgi apparatus out, chances are you won't be able to have hormones leave the cell into vesicles and travel down into circulation because they're going to get degraded in the circulation. So that last little part, you students will struggle with because all they have memorized is that the Golgi is part of protein vesicle transport, and they don't know, they can't understand what happens beyond that. If you were to listen to the material, write it down, and then think about it and spend time with it, I think people would get a higher yield out of the material, and you would start to see improvement on your grades. I think that's essentially my approach. Yeah, Jessica, that's pretty good too. You print out the PowerPoints and then write your notes next to the slide. You have to. The key word here is writing it out. Sitting there and staring at it with memorization, yeah, it'll work, but I don't think it's the best. I don't think it's the best. And then try not to say distracted or trying to avoid any kind of distractions. If you're over there getting distracted, either looking on the internet or your phone, and you're not really focused in, then you're not doing yourself a lot of good either. Yeah, Alyssa, you do have the synthesized material, and then kind of what I think you mean by that is connect the dots. I've used that term quite a bit, where you start connecting the dots. Yeah, and that helps you start to see the big picture. And when you connect the dots, you have to spend time with it. You have to find time. And this class is very demanding. I mean, it has usually there's been past semesters, it's been a 60% fail rate. And that's why we're trying to do these spotlight sessions to help students. But this is this. Sorry, I think my microphone keeps cutting out. This is the same material that's been in the class for semesters on end. So I think that is my, without rambling on any further, I think that is my advice and guidance to succeed in Bio 137. And it really takes something you have to do it every day. I think it's an everyday. This is an everyday class. And there's some classes where you'll have every day. There was a biochemistry class that I wanted to get an A in, and I had to work very hard to get it. A hard-earned A, as I called it. All right, so let me catch up on some of your all's chats here. Okay, Karen said something about the cellular level. I'm assuming you're talking about like organelles and replication. Okay, chemistry, like you're talking about like polar bonds and atoms and that stuff. Atomic theory. Okay, so not organelles, more like atomic theory. Okay. Okay, both. All right, we can do that. All right. So is there any other comments about the class? Any comments like that? Anybody else having ask any questions? I think that's kind of my whole my perspective on how to succeed in Bio 137. Bio 139 is more anatomy and physiology, and you don't really do atomic theory and biochemistry. It's just straight A and P, anatomy and physiology. Okay. All right. So let's talk. We talked about metabolism first from Meredith. So when and help, you guys need to help guide this conversation if I get off topic. Metabolism basically is glycolysis, citric acid cycle, and the oxidative phosphorylation phase. And essentially what we're starting with is that we're starting with glucose, right? Is when we eat a donut or we eat some bread or anything. I think I had some pasta tonight and some Asian noodle dish. eventually things will get degraded down into glucose, where glucose can enter all these different metabolic stages, and we can garner ATP out of out of this for the cells. And glycolysis, we start with glycolysis, and we have glucose that enters, and then at the end of glycolysis, we have pyruvate, right? And we netted 4 ATP, 4 ATP out of glycolysis. Now, that takes place out in the cytoplasm, okay? Glycolysis takes place in the cytoplasm. When we talk about the citric acid cycle or the Krebs cycle or the CTA cycle, it's all the same thing. What we're really doing is taking this pyruvate molecule that we got out of glycolysis, and we are pumping, we're picking out electrons out of the glucose, and we are putting them on an electron carrier molecule called NAD. And NAD is a molecule that can readily take on an electron. In other words, a hydrogen molecule or a hydrogen atom. And this is what happens when the electron gets taken in to the NAD plus. It's called NADH. And what the purpose of this is to take NADH and we stick it in the oxidative phosphorylation phase. And that's how we use this NADH, because it's got a lot of energy in it, to make ATP, right? So we have ADP, adenosine diphosphate, which is really nothing because it has to be adenosine triphosphate. So we use the NADH that we made in citric acid cycle, and we expel that energy to put a third phosphate onto ADP, okay? And that makes ATP, okay? Are we following that? Do we need to go into more depth, or do we need to pull back on the reins a little bit? I'm not sure how deep we went into metabolism. Let me know something. Okay. Okay, Karen gets it. Does anybody else get it, or is everybody else confused, or do we need to? Okay. All right. So this citric acid cycle and oxidative phosphorylation takes place in the mitochondria. That's the powerhouse of the cell. This is where we get all this ATP from. Okay. Now, unless there's any other topics about metabolism, I'm going to move on, but let me know if we need to talk more about metabolism that I've skipped over. Okay. Somebody else mentioned chemistry and atomic theory. Chapter 2 and 3 are always very tough. And basically what it is that when you have atomic theory, this basically surrounds. Okay, cool. I'll put it out, and by the way, that brings it. Meredith brought up a good point. I'm going to throw this up on my YouTube account, so you can check it out later if it's just for accessibility purposes. I think some of you all have been really using it, and I've gotten some good feedback, so that's good. Now, atomic theory basically is talking about protons, neutrons, and electrons, right? And this is what makes up matter. This makes up everything. And the big thing that we're really concerned about, at least in biochemistry and metabolism and biology, is the flow of electrons. And electrons just pass, they go everywhere. And we try to track them. And just to go on some basic atomic theory facts, we have protons that are positively charged. We have neutrons that are neutral charged, and electrons are negatively charged. And those are the ones that really help carry out biochemical reactions is the flow of electrons. And I'm trying to think what else I need to go over. These can be applied to bonds. We talked about polar and nonpolar bonds. This is the chemistry aspect. So if we have polar bonds, that basically means that these bonds can apply to water. They can bind to water. They are water loving. And then nonpolar bonds do not bind to um, do not like water. They're like more lipid based, fat based. Okay, so when we have nonpolar bonds, like it's like oil and water, right? If we shake up oil and water, what happens? Well, eventually it's going to separate, and that is basically a demonstration of polar bonds binding to other polar bonds and nonpolar bonds binding to other nonpolar bonds. Like dissolves like. Okay, and rarely will you see a polar and nonpolar bond near each other. Do we need to talk about covalent and ionic bonds or hydrogen bonds? Okay. Okay. Alrighty. So hydrogen bonds are really interesting because hydrogen bonds are a part of DNA replication or the DNA structure. DNA helps. They are bonded like when we have A and like adenine and thymine bind together, and guanine and cytosine bind. They're really binding through hydrogen bonds. Okay, and hydrogen bonds are basically hydrogen atoms binding to other hydrogen atoms, other oxygen atoms, or other fluorine atoms. So you're going to have HF, which is hydrogen, fluorine, hydrogen, and oxygen, or hydrogen and hydrogen. So HOF are basically the elements that hydrogen bond happens can occur, and they're relatively strong. They can be in numbers. They are seen in DNA replication between the nucleotides. That's what I think A and T have 2 hydrogen bonds between them, and then G and C are a little bit stronger because they have 3 hydrogen bonds between them. All right, and this is what causes the double helix, the coiling of the double helix. Partly is from hydrogen bonds. Okay, so now we've talked about hydrogen bonds. Let's touch on ionic and covalent bonds. Ionic bonds are bonds that do not share electrons. Remember we talked about we're always trying to track electrons in the biochemical world. Electrons are, depending on the bond that we form, really dictates what type of bond it is. Okay, and what I mean by that is ionic bonds. There are certain elements or atoms that will be more electron loving. There'll be other atoms that are ready to give these electrons up. Okay, so let's go back to the periodic table. On the left-hand side, you'll see like calcium, sodium, lithium, potassium. All these are atoms that are ready to give up an electron, because what they want to do is that they want to satisfy their octet rule, right? This octet rule where you have to have a total of 8 electrons, or you want to be balanced. And sometimes you'll see in the real world, you'll see like Na plus or calcium 2 plus or magnesium 2 plus, potassium plus, and that means that they like to give up their electron. So that's why they have a positive after it, because they are positively charged. Okay, they are known as a cation. If you talk about ions, what you're really talking about are these atoms that give up electrons that allows them to become charged. Cations are positively charged ions, And then anions are negatively charged ions. And these are elements, okay? This is like sodium loves to give up an electron, so it's always going to be Na plus. All right, and that is in the first column on the left-hand side of the periodic table. If you look at the second column, the alkali earth metals are the first column. The second column is the alkali earth metals. This is calcium, magnesium. Those are the two big ones, really. And they're always going to be 2 plus because they like to give up 2 electrons. And where do these electrons go? Well, they normally go when these atoms bind to another element, okay? So let's focus on the right-hand side of the periodic table. Fluoride, chlorine, iodine, bromine. These are called halogens, that column. And they are electron-loving, electronegative atoms. So they like to garner electrons because they have 7 electrons and they need 1 more to satisfy their octet rule. Okay? So you will, all the time, you will see chloride minus or fluorine minus. That's because in the real world, if they're just hanging out, they're going to pull the electron from somebody just because they're greedy. Now, if you have, like, let's go back to sodium. I mentioned sodium is +1. I also mentioned that chloride is minus all the time. So chloride loves to pick up an extra electron. That's why that's chloride minus. Well, it just so happens that sodium loves to give up an electron. Hence, that's why you have NaCl. Okay? They go together. That's salt. And that is what we would call an ionic bond because there is no sharing hardly of electrons. It is chloride takes the electron from sodium and creates this bond, and that's how you have an ionic bond between sodium and chloride. Covalent bonds share electrons, so it's not exactly one ion domineering the other ion like you have in chloride and sodium. So covalent bond is sharing electrons, and then ionic bonds are kind of somebody's being more dominant than the other with the atoms. You guys are going to have to forgive my animals back here. I guess they wanted to come in and see who I was talking to. Okay, so those are bonds. How do we feel about that conversation? Are we okay there? Do we need to clear up some stuff or tie down some loose ends? I felt like that went on for 10 minutes. Okay, well, thank you, Jennifer. Okay. Okay. So what else did we talk about? We mentioned organelles, right? Organelles. Okay, you guys might have to help me with this depending on which one you want to talk about. So what is the powerhouse of the cell? Let's just start off with that. We've already mentioned it before. That's right. This is where, yeah, cool. Everybody's coming in. Good. So, all right. Now, that is metabolism. That's where we're going to get our ATP. Okay? That's important. Now, endoplasmic reticulum. There's 2 types. There's a rough and a smooth. And basically what endoplasmic reticulum does is that it can modify proteins. It can stick signal molecules on it and do all types of different things. There's a smooth and a rough endoplasmic reticulum. But let's move on to Golgi, because Golgi is more important in packaging up proteins so they can be excreted out into the the circulation, right? This would be some type of water soluble protein that needs to be packaged in vesicles and then released out into the extracellular fluid to go travel to somewhere else. Sometimes when we have hormones that get released into the pituitary gland up here, they have target tissues that they will go and visit to carry a message down into like the kidneys or the adrenal glands. So they have to be properly packaged for the long haul from your pituitary gland down to your kidneys. And your Golgi apparatus helps you accomplish this. Nucleus, we all may know, that's kind of like the brain of the cell. That is where the chromatin is. Chromatin is just a fancy, it's not a fancy, it's when we talk about, I think somebody talked about earlier, like everything at the cellular level. Well, if you want to go down to the cellular level, we're really talking about like nucleotides. That's about as small as we can get with DNA, right? So we have nucleotides with DNA making up these strands of DNA, right? Parent and complement strands. And then, excuse me, heart being. And then after the DNA nucleotides, we zoom out a little bit more and we start to see these chromatin shapes. And what that is just DNA being packaged around these histones to eventually form chromosomes. Chromosomes are huge. Chromosomes, we're zooming out just a little bit more. And what happens is that when we're talking about chromatin, we're just talking about wound up DNA. And then if we zoom out a little bit more, this chromatin will make up the chromosomes. Chromosomes are found in the nucleus. And so it's kind of important when you hear about all these vocabulary words that you really know where, like somebody talked about everything at the cellular level. Well, the cellular level is going to be probably, chromatin or DNA strands. And if you want to get down to like the molecular level, like hardcore microscopy, then you need to get down and or like, any kind of like gene testing, and you're talking about nucleotides. Right. So it's good that you guys are kind of asking questions about this because this helps determine it. How far detailed are we talking about? Are we looking at like a bird's eye view, which is like chromosomes, like big chromosomes inside the nucleus? Or are we talking about individual nucleotides and how DNA is replicated on like a molecular basis? So I always struggle with that when I was in your all seats. Like, what in the hell are they talking about? Are they talking? It just confused me, and you have to really sit and think about it. Same with proteins, like amino acids and proteins. Amino acids make up the protein, and then the protein can fold up and create these like large intricate enzymes that carry out these biochemical reactions. All right, so what else do we need to talk about? Like we talk about ribosomes. Ribosomes help are the devices that are floating around in the cytoplasm that help create proteins, amino acid chains, polypeptide chain, as people call it. That's the same thing as a protein chain. Are there any other organelles that I'm missing? I don't think so either. Okay. All right, so what else do we need to talk about? Okay, tips for the current lab. The quiz. The quiz, let me go pull it up. Because this one's hard because it's on histology. So it looks like a lot of these pictures are going to be coming straight from the lab manual. Okay. So we have 10 questions, and they're going to have these arrows, and they're going to say, name the tissue indicated by the arrows. So this is going to be a picture straight from your lab manual, and it's going to show you what it looks like. Okay. Now, that's number one. Name the tissue indicated by the arrows. And then it's going to tell you, name one location of this tissue. So you have to know the locations of the tissue. And then the question 3, name the tissue indicated by the arrows. All right, and these are all from the lab manual. This all is it's some kind of tissue. It's not connective tissue. It's some kind of non-connective tissue. I think there's a few questions about connective tissue, like at the end. And then it says name the function of one of these tissues. So be sure you are able to identify what it is, where it is, and the function. It's kind of important. Number 5 gives you an example of, it's either cilia or microvilli, and it says name the tissue indicated by these arrows. Okay, and it's either pointing at some kind of goblet cell, microvilli, or cilia. You know, the important features that we discussed on each one of these tissues, especially if you had me for lab. I think I hammered on these pretty hard last week. Number 7, it says name the tissue indicated by these arrows. I think that is a connective tissue of some sort. Number 8 is the same thing. What type of fibers is indicated by the number 2 arrow? So when you talk about connective tissue, make sure you understand the fibers. I think the answer is actually elastic fibers, but check me on that. Number 9 gives you a picture of what type of connective tissue this is. And then number 10 is the location of this kind of connective tissue. So that's the quiz. We got any questions about that? Yeah, I think that's helpful. You're welcome. All right. Is there anything else we want to talk about? Yeah, Farah, it certainly does. So when we have, and I'm going to try to explain this in the simplest words as possible, because we could spend an hour on DNA replication. When you have DNA replication, DNA polymerase can only move from 5' to 3', right? So the easy part is making the leading strand, where it literally just complements the parent strand and will replicate it just down the line, 5' to 3'. However, the other strand that is being made is opposite. It is from 3' to 5' in that direction. So what has to happen is that DNA replication has to go backwards. It has to like let's say it goes like this is from 5' to 3'. 5' over here, 3' over here. So DNA polymerase can go this way, and then all of a sudden it has to break, and then it has to go back way back over here and then do a little bit more and then stop, and then go way back over here and then go and stop. And it creates these fragments, okay? These fragments, and those are these Okazaki fragments that has to happen. And there is another enzyme that comes in that's called ligase. And ligase will connect these Okazaki fragments together. So it's a continuous strand. But yes, it is more work, and DNA polymerase has to backtrack and go over the lagging strand. It's called the lagging strand. Does that help clear things up a little bit? It all has to do with 5' to 3' movement. Cool. Alrighty. Okay. Any other topics? I'll get you guys the attendance sheet if you think we're good. And I think we're going to have our first lab exam coming up in 2 weeks. So we'll probably go over that a lot more too. All right, so this is the new attendance form. Let me know how that goes for you guys, and I'll hang out. Yeah, I agree, Karen. I think make sure you schedule something either for me or Dr. McCain. I don't know how much in depth you'll go into the exam. I'll try to give you guys some high yield material to make sure you go over. So yeah, make sure you get your spots for that because I'm assuming next week's gonna be busy. And then some of you all have to, since I got a bunch of you all in here from different areas, if you are doing the online labs, check your email. I highly doubt I'm having lab on Wednesday night, just because given the weather coming in. So if you're one of my people or you do have an in-person lab, you might want to check your email to see what we're doing because I'm not getting out in freezing rain. No way. I'll be lucky to make it to work. So FYI on that. But do the attendance form and let me know when you're done, and then that's all we have prepared today, unless we have other topics that we want to talk about. Thank you, Kendall. If you got it all figured out, you're good to go. No problem. That's what I'm here for. Guys, come back. Make sure you got questions or topics you want to talk about. It makes things go by a lot faster, and you're able to get as much as you can out of these spotlights. No problem. Anytime. All right. You guys be safe this week. It's going to be some bad weather in the midweek. Make sure you're okay on those roads. Freezing rain's hard to deal with. All right. Looks like everybody knows everybody's out of here. Okay. All right. Thank you, guys.